Hello, students. This is Dr. Faizan Mirza. I'm discussing with you a question where we are dealing with chi-square test, and I'm discussing a few questions regarding inherited change as well here. So when we're discussing this inherited change, I know when many students face difficulty here. So the question given to us is that there's a fruit fly, dorsophilla, melanogaster. You know this is the species name. This is genus. It feeds on sugar found in damaged fruits. This is some information given to you. A fly with normal features called wild type. So if there are normal features wild type, so this is the wild type given to you. Just keep on relating to what's ever mentioned, and you keep on following with the diagram as well. That what actually is meant by each word here. It is having a striped body, and its wings are longer than its abdomen. So you can see that its wings are very long as compared to its abdomen length. So this is mentioned to you, and it has a striped body as well. So you can see the stripes on its body as well. Furthermore, there are mutant variations as well. So mutant variations mean the ebony and vestigial both are mutant variants. So these both are mutants, and this one is wild type. So the mutants include ebony body colored. Ebony means black, so this is ebony body, and ebony body or vestigial wings. So what the vestigial wings are, these short wings which are not very long and not longer than the abdomen. So this all information is already mentioned to us. Now the further they're saying wild type features are coded by dominant alleles. This capital A for wild type body and capital B for wild type wings. So we just know that if the B is having the wild type body, then definitely it means it will have at least one of the capital A. So wild type body will always have at least one capital A, the dominant allele. Similarly, the wild type wings are these large wings. If even a capital single B is present, then that means the wings will be of wild type, which obviously means that ebony body and the vestigial wings are recessive alleles. So this is A and this is small b. So there are further they're asking explain what is meant by the term allele dominant, alleles and dominant allele. So allele is just a variant of a gene. You just need to recall fact and just answer that. So it is a variant of a gene that is having the same locus on the homologous pair. So it's always present on the same locus on the homologous pair. So you just mentioned that. Dominant, it is the allele that is always expresses itself in a phenotype and is present in the genotype. So irrespective whether it's there or not, dominant will always express itself. So the information that's given to us, actually this tells us that, for example, if it's capital A, small a, both of them will have striped body. So I just prefer to annotate my paper whenever I'm solving. So capital A, small a, they both will come for striped body. So this individual will always be any of these capital A, small a. Whereas ebony body will always be small a. Small a, we decoded this information from the information which was already given to us in the question. Similarly, come to wings. Capital B, small b. This is represented in the wings, the long wings, and vestigial wing is small b, small b. So the wings present here, vestigial wings are small b, small b. So keeping that in mind, we can just write these letters to just understand how this basically occurs. Mutant variation means we know whenever a mutation occurs, by chance it could occur, a nucleotide sequence will be changed, and the gene will end up having a new allele just by chance. Next, they are stating two wild type fruits were crossed. Two wild type fruit flies were crossed. Each is having allele A and B and carried alleles for ebony body vestigial wings. So A and B but were there in both, and they carried small a, small b as well. So we know that when that's happening. So first of all, we'll just write the genotype and the phenotype of the parents. So the parent genotype will be capital A, small a, capital B, small b. This will be the genotype because it's mentioned to us that it carried capital A but with a small a as well. Similarly, it carried a capital B with a small b as well. So we know that the genotype will be capital A, small a, capital B, small b. The same goes for here as well. Now, what does this mean? If even a single capital A is present, then definitely the body is striped. So we mentioned that it's a striped body. And if it's a single capital B, so it's having long wings. So two of these varieties are crossed. So first, in the first row, we mentioned the parent genotype. In the next row, we mentioned the parent phenotype. The parent phenotype in the first row, and the parent genotype in the next row, and then we go for the gametes. The gametes, we know whenever we are making gametes, we just split the homologous chromosomes apart. This AB, this A and this A, this represents a homologous pair. Similarly, B and B, this represents a homologous pair. So whenever we are making a gamete, we need to split a homologous pair. So in each of these four gamete, if we are converting this homologous pair into a gamete, we cannot place both these in the same gamete. So of capital A and small a, either one will land in one gamete. Both cannot land together. So we'll make two gametes with capital A's and we'll make two gametes with a small a. The same goes for BB, capital B, small b. So we need to split this as well. So one goes with capital B, the other goes with a small b. Here one goes with capital B, the other goes with a small b. So these are the four possible variations of the gametes that can be produced. The same goes for here. So we have four types of gametes here. We'll make a dihybrid cross. We'll just mention the gametes of each at the top. So this AB is mentioned here, this maybe is mentioned here, this AB is mentioned here, and finally this AB is mentioned here. Similarly, we have this AB here, this AB here, this AB here, and this AB here. And then we make a 4x4 box, and this 4x4 box actually is having all the places in which we'll place the genotypes of these organisms. And once the genotypes have been placed, we'll start to just write down their phenotypes. Whenever we have mentioned the phenotype, it's better to write the phenotype here only. So if for even a single capital A, we just write striped body. We know even if single capital A is present, that means striped body. And wherever we have small a's together, that means the body will be ebony. So we can mention ebony body there. Wherever you have small b's together, that will be vestigial wings. So just vestigial wings and so on and so forth. So there are a total of 4 different type of organisms, phenotypes which are being produced here. So striped long, striped vestigial, ebony long, and ebony vestigial. If we just start to count these, our count comes out to be 9331. So 9331 means that the ratio is 9331, which means that the allele A, the gene A and gene B, they both are following independent assortment. Whenever independent assortment is occurring, it means that new kind of gametes are formed and recombinant can be seen. So in this case, we know which can be recombinants. If we try to recall what recombinants are. So if the parental phenotype is striped and long, we know striped was present with long. So any of these which having striped long will actually be the phenotype of the parent who's non-recombinant. But striped vestigial and ebony long, these are the two which will be in this case the um, recombinant ones. Next, you're stating you, okay, what we are doing, we are having the two heterozygous flow flies which are crossed 384 eggs hatched and developed into adult flies. The formula for chi-square test is given to you. In chi-square test, we have observed and expected. And for example, if I was asked to write down the null hypothesis in this question, what will be the null hypothesis? So what will be the null hypothesis here? I would state that there is no significant difference in the observed and the expected ratios of the cross between two heterozygous these fruit flies. So that will be my null hypothesis. So what I'm doing, I have my observed numbers given to me that out of 384, these individual 384 eggs were having these numbers of observed. Our expected ratio will be 9331, which we have already discussed. So 9331, as far as 9331, my values which will be expected, I can take out 9331 ratio, and using this, I can calculate our values. I've discussed chi-square test in one of my detailed videos further as well, previously as well. So then we calculate O minus E. When we are calculating O minus E, so this is O and this is E. So what you'll be doing 79 minus 72, and the answer will come here. Now, once the answer is here, the next thing is we do the square of this. So this is 7 squared. So this comes here. Then the last is O minus E squared over E. So the squared answer, which we are getting here, 49, I'll divide it by E. My value of E in this case was 216. So I'll use my value of E and I have my answer here. Finally, the chi-square test. So this is the chi-square test. In chi-square test, we take the sum of O minus E squared upon E. This is O minus E squared upon E. I need to take the sum of this entire row. So I'll take the sum of this entire row because this row represents to me O minus E squared over E. I need to calculate the summation of this. So if I add all these values together, the last column, my answer comes out to be 2.78. Now, if my value is 2.78, what I'll do next, I'll go and calculate my critical value. The table is given to me, and as they are saying, as 4 transfer data was there, the degree of freedom was 4.3. We know that degree of freedom in chi-square test is a number of the categories minus 1. So number of categories of 4, so we subtract 1 by 4, 1 from 4, so we get 3. So the values are given to you, probability values. So we'll use a value of 0.04, probability greater than, because this is what we use in all biological experiments until otherwise stated. So our critical value is 7.82. So this is the critical value. We have to compare this critical value with our calculated chi-squared value. So calculated chi-squared value was 2.78. We can see that the calculated value of chi-squared is very small as compared to the critical value. So the difference between observed and expected is not significant. It's only due to chance. And what do you mean by not significant? For example, we expected these to be 216, but they came out to be 207. Yes, the value is different, but not significantly different. Similarly, if we expected 72 here, but we got 79. So again, the value is different. We expected a different value, we got a different value, but is this difference significant or not? No, this difference is not significant, it's just by chance. Here we expected 72 for ebony body and long wings, but we got 68. Again, there's a difference, but the difference again not significant, it's just by chance. Similarly, I expected ebony and vestigial to be 24 out of these 388 or the 384 given to me. So I expected 24 of these to be ebony vestigial, but I got 30. So out of 24, I just got 30 in observed. So yes, again, the difference is there, but the difference is not significant. It's only due to chance. So my conclusion here, they are asking explain whether or not the observed results were significantly different from expected results. So chi-squared value is smaller than the critical value, hence the difference between observed and expected is not significant. It's merely due to chance with 1 in 20 chance of error. Why I'm mentioning 1 in 20 chance of error? Because this is what is represented by probability greater than 0.05. So whenever we are using probability greater than 0.05, we mean that there's the chance of error is 1 in 20 chance of error. That's it from my side. Thank you so much.